Hello, you are welcome back to Unit 6, Section 4. Under this session, we will be looking at dehydration, which is another important stage in histology. In this session, we will discuss the need for dehydration and how it is done. At the end of this session, you should be able to explain the need for dehydration in histology and describe the procedure for dehydration, and finally explain the need to dehydrate gradually. Hope you enjoy this session too. In histological procedure, there is the need to dehydrate the tissue in that this particular step helps to allow for complete infiltration of tissues with Canada balsam, which will be used later in the preparation of the tissue for examination. If this particular procedure is not properly done, it could cause incomplete infiltration and the tissue will appear opaque, and also the tissue could be subjected to bacterial decay. Dehydration procedure, that is removal of water, should be done gradually, and there should be a sufficient time that is allowable for the complete extraction of water. And if it is done rapidly, it will cause distortion and shrinkage of the tissue by setting up violent diffusion current, especially on delicate tissues. The common method in dehydration procedure is to successively pass the tissue through a series of alcohol or ethanol with increasing concentration, in that at the end you immerse the sample 2 times in an absolute alcohol. The detailed procedure of dehydration involves carrying the tissue together with their identifying labels from one container to another at the appropriate times, and this allows them to drain for a few seconds on blotting paper between each change. If the tissue is more delicate, normally alcohols of lower grade are suitable for commencing the dehydration process, and the smaller the interval, there should be between the strengths of the ascending alcohols as well. And in order to minimize tissue distortion resulting from diffusion current, specimens that are delicate are dehydrated in a graded ethanol series from water through 10 to 20%, 50 to 95%, and 100% ethanol. Also, in summary, the strength of the initial alcohol that will be used in the dehydration procedure and the time required in each grade will depend on the size and the type of tissue, as well as the fixative which was used. In the paraffin wax method, dehydration procedure normally begins by making use of 60 to 70% ethanol or 90 to 95% ethanol, and then 2 or 3 changes of absolute ethanol. The duration of the dehydration should be such a way that it is kept in the minimum, which will be consistent with the tissues being processed. The tissue blocks, which are 1 millimeter thick, are normally kept in 30 minutes in each alcohol, whilst for 5 millimeter thick, they are kept for 90 minutes or longer in each change. One thing we have to note about the use of ethanol is that tissues can be held and stored indefinitely in 70% ethanol without causing damage to the tissue. Now have a look at these questions. In histology, what is dehydration? Why should dehydration be gradual? Which reagents are used in dehydration? Under Section 4 of Unit 6, we studied about dehydration of tissues and why it is important to dehydrate tissues in histological studies.